Hello everyone, myself Professor Nandita Bhakat from Department of Botany, Narajole College. So today in this session we will be discussing about splicing, the splicing mechanisms. So before starting the topic, let us discuss that what is splicing and where it occurs and what is the necessity of it, right? So what is splicing? Now, in eukaryotic cells, what happens immediately after transcription, the RNA that we get is called hnRNA. Okay, so that RNA undergoes some modifications. So three types of modifications are there, among which one is splicing. What happens in it? Now, in this, the introns, which is the non-coding part or the non-coding sequences, and the exons, which are the coding sequences, which are present in the hnRNA. So the introns are removed and the exons are joined together. This is called splicing. Okay. So why are these introns removed? Because they are non-coding sequences, means they are not involved in protein synthesis. But the exons, they are coding sequences and hence they are there for future protein synthesis, which we can see in translation process. Okay. So now the question is there, one thing, that if introns are of no use, okay, then why are they in the RNA, right? So because everything is there, means there is a reason it is there, it exists. Okay. So there is also a reason why introns are there, and the reason is for the polymorphism. Okay. We see a variety of features, characters among the population. This is all because of polymorphism. Okay, so we'll come back to later. And now we will be discussing about the splicing mechanism. Let me first tell you that in splicing there are two main types. Okay. One is self-mediated, means the introns, they are spliced out from the RNA without the aid of any kind of protein or other RNA molecule. That is self-splicing. Another one is spliceosome machinery mediated. Means here there are certain types of RNAs and proteins which combine together to form a complex molecule that is called a spliceosome, which helps in the process of splicing. Okay, so let us see. First, we will draw the image or the structure of the intron and so on, and we will define the various terms that are involved here. This is the 5' site on the 5' exon. And this is the 3' exon. This checked region is the exon part, and this is the intron part. This is the exon part, and in between this is the intron part, which will be spliced out. You see the mechanism, the chemistry behind it. Okay. Now, let me draw the picture first. 3 prime splice site and this is the 5 prime splice site. Okay. Now, let me tell you that these are the borders. This is actually, we will study about the chemistry of RNA splicing. These are the borders. This is the border of exon and intron, and in this 3 prime region you will also see the border between the exon and the intron. They are marked by specific nucleotide sequences like GU, AG sequences. It is also known as GTAG rule. Okay, GTAG rule. Means this rule is present in the border region. It indicates the border between the exon and the intron, GTAG. Now the 5 prime end of the intron, it is marked by the 5 prime splice site. This is the 5 prime splice site, and this is the marking the end of the 3 prime region of the intron is the 3 prime splice site. There is a branch site, branch point within the intron. Within the intron there is a branch site, and it is followed by pyrimidine track. Why? It stands for pyrimidine polypyrimidine track, that's a PY. Now there are some conserved sequences, that is the GU in the 5 prime splice site and AG in the 3 prime splice site. These are the conserved sequences, and A in the branch site. So GU, AG and A, these are the conserved sequences, all are present in the intron. Now the intron, it is removed by 2 successive trans-esterification reactions. Okay. Now let us draw that picture where we can find the trans-esterification reactions. Hopefully this one was clear with you all. Okay. Now we will be studying about the chemistry which is directly involved for this splicing mechanism. Sorry, this is the branch site. This is the exon part. So this is the intron region, and this is the exon part. Okay. Now this branch site acts as a nucleophile, and this touches with the phosphate group of the guanine. As a result of which, what happens? This bond breaks down at this border site. The bond breaks down and the exon becomes separated. It looks somewhat like this. This is that branch point. Actually, when this bond breaks, the axon becomes separated, and this it turns and again bind with this branch point. Okay, it forms a lariat over here. After that, what happens? Another esterification reaction it will occur. That is, the phosphate over here with the guanine and adenine. Now this OH it will act as a nucleophile and will bind with this phosphate and will break the bond over here. Now as a result, this axon will become separated. And as a result, what forms up at the two axons, okay, they are joined together and the lariat it is released, and the intron it is released as a lariat, and this is the axon. This is the basic chemistry which occurs over here. Let me discuss it once again. So what happens that the introns, they are removed. This intron is removed by trans-esterification reaction. What is trans-esterification reaction? In here, what happens? One ester bond is formed and the other is broken down. So the first reaction triggered by the 2' OH, 2' OH of the conserved A at the branch site. This group acts as a nucleophile. It acts as a nucleophile, and it attacks the phosphoryl group of the conserved guanine in the 5' splice site. This is the 5' splice site, and this is the 3' splice site. So this nucleophile will attack the phosphoryl group of the guanine of 5' splice site. The freed 5' end, this is the freed 5' end of the intron, is joined. This is the freed 5' end of the intron. This one, this portion, it joins with the A, that is the branch site. This is one esterification reaction. In the second esterification reaction, what happens? 5' axon. This is the 5' axon. Now it will become or act as a nucleophile and attaches the phosphoryl group at the 3' end. This is the 3' end on the 3' splice site. It will attach with the phosphoryl of the at the 3' splice site. Okay. This results in liberating the intron as a lariat and joining of the 5' axon and the 3' axon. This was the 5' 3'. The axons become joined together and the intron is released as a lariat. So, as I was discussing previously, that splicing occurs in two ways. One is self-splicing and one is lysosomal-mediated. So this is self-splicing. Okay. Self-splicing is again two types. Group 1 intron and group 2 intron. Okay. This one was group 2 intron. Okay, this is group 2 intron. In this type of splicing, we will find the lariat. Release of the intron in the form of lariat. Now we will discuss about the group 1 splicing. Group 2 we have discussed. Group 1 we will discuss. Okay. Here actually it is a formation of guanine pocket. We will be discussing about that formation of the guanine pocket in group 1 splicing mechanism, which was absent in group 2. This is the guanine pocket. Okay. And this acts as a nucleophile. Rest all the conserved sequences or the that is a splice site, and these are all same. Okay, but here we find a specific guanine pocket. Now what happens? This guanine, which will act as a nucleophile, it will attach to this region, this boundary, intron-exon boundary of that the 5' splice site. Let me show you in the picture form, it will be better. Bedroom. So this is the basic concept. What happens here? Now the RNA, it folds in a manner to form a guanine binding pocket, which allows the molecule to bind to a free guanine nucleotide. So this is a pocket which will allow guanine to bind to it, a free guanine to bind in this pocket, and use that to initiate the splicing. Okay, so when this is acting as a nucleophile, it will break the bond over here at the 5' splice site branch, and after that the hydroxyl, this 5' splice site region will act as a nucleophile. It will again break the bond over here at the 3' splice site. As a result, these two exons will be joined together and this intron will be released or removed. But here we are not finding any kind of lariat formation. Okay. So this is the difference. One we are finding a lariat and here no lariat, and that guanine pocket is present in group 1 intron, which is absent in group 2 introns. Next, we will study about another type that I was discussing you about. That was the non-self splicing. Means it is not self-couplings. It is mediated with the help of different type of proteins and RNAs. They are composed together to form a spliceosome, a big molecule spliceosome, and with the help of that spliceosome, splicing will take place. So let us now study about spliceosome machinery. The spliceosomes are also mediated through molecular machines called spliceosomes. Okay, the molecular machine is called spliceosome. This complex, it comprises of near about 150 proteins, near about 150 proteins, 5 RNAs, and this together its size, it's like a ribosome. It's such a big like a ribosome. It's similar size to a ribosome. So the 5 RNAs. What are these 5 RNAs? These 5 RNAs are U1, U2, U4, U5 and U6. These are the 5 RNAs, okay, which are involved in this spliceosome mediated splicing machinery. And these are collectively, these 5 RNAs, they are collectively known as snRNA. snRNAs. And these are also composed of serotype proteins. Means this proteins plus RNA. Now the RNA protein complex, this is called the snRNA complex. So this complex is called the snRNA complex. Spliceosome is made up of snRNAs, snRNPs. Okay. Different type of snRNPs come and go at different times, performing specific functions. So snRNPs means small nuclear ribonucleic proteins. The snRNPs, they have 3 roles in splicing. snRNPs, they have 3 roles in splicing. What are these 3 roles? The first, they recognize the 5' splice site and the branch site. Okay, the first is the recognition of the 5' splice site and the branch site. What is the second role? Second role is to bring these sites closer. Bring these sites closer as required. And the third is to catalyze the RNA cleavage. Catalyze the RNA cleavage and join. Okay. So these are the 3 roles of snRNPs. Now, apart from these, there are some non-snRNPs are also there. Non-snRNPs are also involved in splicing mechanism. So these are the functions of the snRNPs, and there are some non-snRNPs which I am telling you. There are 2 main non-snRNPs which are involved in splicing mechanism. One is U2AF, that is the U2 auxiliary factor. Okay, so the function of this U2AF is to recognize the polypyrimidine group that we have studied, that Y, it is denoted by Y, PY. It recognizes that unit, and another one is BDP, that is the branch point binding protein. Branch point binding protein. Okay, so these are the non-snRNPs. Okay. Now we will see the chemistry with the help of a diagram for a better understanding. Hope this was okay with you all. electron splice site, 3' splice site, these are the splice sites. This is the exon, this is the intron. This is the U1. We studied the, what was the U1? The U1 was the RNAs. We find different type of RNAs over here. U1. And this is the branch site, and out of this branch site BBP will be joining or recognizing the branch site, and that U2F, it has two units. Okay, this is larger unit, this is the smaller unit. U2A6F, U2A65, and this is 35. This is the larger part, this is the smaller part. Okay, next, what comes? This. This is broken out and comes in U2. Okay. In the next step, we will see that this is the 65 larger unit, this is the 35 unit of U2F. Okay, now we can see that different type of RNAs and proteins there, you will see that they are coming in. Someone is leaving out, someone is again joining. So it is a process which comprises of many RNAs and proteins. We will find out. Now a group of RNPs. They will be entering the process. U5, U4 and U6. So we will enter and release of this U2F factor. This will be released out.